Hi, my name is Olivia, and this is my exam mini video 3. Today, we're going to be talking about the features of Watson and Crick DNA. Please pause this video to get a brief description of what Watson and Crick DNA is and a snippet of what's to come. Base pairing between two strands of nucleotides allows for constant width of helix. The basic rule designed by Watson and Crick is that adenine binds to thymine and cytosine binds to guanine, forming base pairs through hydrogen bonding. Purines are one of two heterocyclic nitrogenous bases found in the nucleic acids DNA and RNA. It is a two-ring structure which can be shown by adenine and guanine. Pyrimidines is the second of the two heterocyclic nitrogenous bases found in the nucleic acids DNA and RNA. It is a one-ring structure, and examples in DNA include cytosine and thymine, and in RNA, an example is uracil. Now we're going to be talking about the ribose carbons. 1' carbon attaches to the base, 2' carbon distinguishes DNA from RNA, 3' carbon bonds to the next 5' phosphate, and 5' carbon has phosphate. Hydrogen bonds are relatively weak attractions between positively and negatively partially charged molecules. They are not the main force joining the two strands of DNA. Adenine pairs with thymine using 2 hydrogen bonds, guanine pairs with cytosine using 3 hydrogen bonds. The sugar phosphate backbone is an alternating chain of deoxyribose sugar and phosphate groups in the DNA polymer. Phosphodiester bonds connect the sugar phosphate backbone. Pause this video to see what a phosphodiester bond is. The twisting of the DNA double helix and the geometry of the bases creates a wider gap called the major groove. It also creates a narrower gap called the minor groove. The major and minor grooves are binding sites for DNA binding proteins during processes such as transcription and replication. Double-stranded DNA is an anti-parallel molecule, meaning that it's composed of two strands that run alongside each other but point in opposite directions. In a double-stranded DNA molecule, the 5' end of one strand aligns with the 3' end of its partner and vice versa. Hydrophilic interactions are maximized by base stacking and are the main force in stabilizing the double helix. Pictured on the left shows flat base pairs lying perpendicular to the sugar phosphate backbone. All helixes have a handedness, which is a property that describes how their grooves are oriented in space. A left-handed helix coils up going to the left, and a right-handed helix coils up going to the right. In Watson and Crick's model, the two strands of DNA twist around each other to form a right-handed helix. After being given all that information, you may be wondering what happens to the stability of a double-stranded DNA when it is placed in a solution with a higher salt concentration. Let's find out. DNA is negatively charged because the phosphates that form the sugar phosphate backbone of a DNA molecule have a negative charge. The denaturing of DNA means breaking the hydrogen bonds and hydrophobic interactions with heat or chemically, which allows the two strands to separate. However, DNA at higher salt concentrations has few hydrogen bonds, even at temperatures higher than Tm. Adding salt also neutralizes the negative charges given from the phosphate backbone and leaves more opportunity for the bases to come together rather than separating, allowing for stabilization. Therefore, adding salt leads to the increase of Tm and stabilization of DNA. Now here are some lists of my references.